Good morning. Welcome to the BRSL webinar series. Before we begin, a few reminders. The audience microphones will be muted. If you are having technical issues with audio or video, please message us using the chat box. We encourage everyone to actively participate by asking questions. Just write them on the chat box and we will try to get to as many of them as we can after the two speakers have presented. So we hope that you will stay until the end of both lectures. Also, at the end of the webinar, we will provide a link to the evaluation form that you must fill out in order to get your certificate of attendance. This webinar will be recorded, so those who are unable to attend the webinar today or who would like to watch the lectures again may view the video which will be posted on the BRSL Facebook page. To start off this session, let us first hear a few words from the head of the BRSL, Dr. Pierre-Angeli Vital.
Thank you, Erna. Good morning, everyone. I formally welcome you to the second of the Biological Research and Services Laboratory of the Natural Sciences Research Institute, University of the Philippines, Diliman, or simply BRSL webinar series titled Plant Talk, Listen, We Do Matter, First. As part of the efforts of BRSL to contribute to the understanding of science, particularly biology in the community, of course, we have to be discussing and up to date with all of the biological topics. And I also welcome back again those who have attended the first webinar. I know that we have all been affected by this pandemic, and as an extension service to reach out to everyone, our lab will try to give free biology-related webinars every month. And for this second webinar, we are fortunate to be joined by experts, Professor Pastor Malabrigo Jr., with areas of expertise plant taxonomy, marine botany, biodiversity conservation, and forest restoration, and Dr. Lerma Maldia in tree genetics, including population genetics, phylogenetics and phylogeography, evolutionary biology, and conservation genetics, and they will walk us through their plant worlds, and as participants, you will be given the chance to interact with them through our Q&A sessions later. This webinar will not be possible without the efforts of the wonderful and hardworking researchers and staff of BRSL. And for this webinar, your co-hosts, as you can see from your participants tab, are Erna, Rocky, Rian, Brian, Jude, Jessa, Donna, Serena, and Ryan. The credit, of course, goes to them. On another note, and just to share with you, you might also be interested once we start the new normal of everyone going out. BRSL also offers workshops like Laboratory Animal Workshop conducted in compliance with the Republic Act number 8485, or famously known as the Animal Welfare Act, Basic, as well as Advanced Molecular Biology Techniques Workshop, the Microalgae Workshop, and soon we will have Phytochem and the Natural Products Workshop, and biological services such as cytotoxicity, genotoxicity, and of course, molecular biology assays, among others. So for the meantime, we offer online consultations and feel free to email or contact us if you have biological concerns. With this, I wish you have a productive webinar. And of course, with our great speakers for this morning, I'm very sure you will. Thank you very much, and I turn you back over to Erna.
Okay, thank you, Dr. Vital. And now on to our first speaker. Professor Pastor El Malabrigo Jr. is currently an associate professor and the chair of the Department of Forest Biological Sciences, College of Forestry and Natural Resources, University of the Philippines, Los Baños. He has also been serving as the curator for trees in the UPLB Museum of Natural History since 2012. He is currently in the PhD by Research program at the UPLB, where he also obtained his BS and MS degrees in forestry. His fields of specialization include plant taxonomy, marine botany, biodiversity conservation, and forest restoration. Professor Malabrigo is a very prolific researcher and writer. To date, he has already authored 104 scientific papers, including 5 books on Philippine native trees. He is the author of the book Shades of Majesty, 88 Philippine Native Trees, that won the 2013 National Book Award and the 2014 Gintong Aklat Award. He co-authored the book Biodiversity Assessment and Monitoring System for Terrestrial Ecosystems, which serves as the national standard for all protected areas in the country. He is an internationally recognized plant taxonomist, the only Filipino member of the Global Tree Specialists Group and the Species Survival Commission of the International Union for Conservation of Nature. Currently, he is leading the IUCN Red List assessment of 800 Philippine endemic trees. Today, he will talk about the diversity and conservation status of Philippine native trees. Let us welcome Professor Pastor Malabrigo Jr.
Yes, thank you very much for the introduction. Okay, I'll share my screen. Okay, so thank you all for coming and joining this Plant Talk webinar series. And of course, special thanks to the organizers for inviting me as a speaker for this talk. Okay, in the interest of time, I'll go straight to my presentation. So this slide summarizes the biodiversity richness of the Philippines. But for this talk, I will only discuss plants. It's a plant talk, specifically trees. Philippines, despite the very small land area that we have, is the 25th most plant-rich country in the world. I assume many of you have visited or have used this online database for Philippine plants, better known as the Coast Digital Flora of the Philippines. This is becoming the most popular and readily accessible reference for Philippine plants. But I suppose not many are aware that all the little details about every species, genus, and family on this database were products of long years of hard work of our very own, the greatest Filipino botanist, the late Leonard Coe, the man in the photo. Leonard was my great mentor and a dear friend. I can say Leonard was 6 months short to his publication of a book that will replace the Merrill's enumeration of Philippine flowering plants. He really wanted to have a comprehensive and updated reference on Philippine plants. And every one of Leonard's friends, including myself, had the e-copy of the world files of this database. Few months after he died, his database was published through a website named after him, Coast Digital Flora of the Philippines. Unfortunately, he was not even considered as one of the authors of his online database. Anyway, according to Leonard's database, we have more than 10,000 species of vascular plants, 89% of which are angiosperms, 10.5% are ferns and allies, and only 0.5% are gymnosperms. And of the 9,432 native species, 46 species or 46% are endemic to the country. Okay, so out of the more than 10,000 species of plants, how much are trees? So the most comprehensive reference on Philippine trees is the book Lexicon of Philippine Trees. The first revision was authored by Felipe Salvosa and was published in 1963. In 1998, a revision was published by Rojo. So it listed 3,084 native trees, of which 800 were classified as endemic and 930 are not endemic or non-endemic. However, 1,355 trees have no information on distribution, and therefore endemicity cannot be determined. So this book, of course, does not include species described in 1998. But aside from that, we have found some crucial errors that the author may overlook. The most serious one is on the genus Saurauia. There is only one species of Saurauia listed on this book. But the truth is, we have more than 50 species of the genus. That actually resulted to the wrong notion, particularly among foresters, that all the species of Saurauia is Acola labang or Saurauia latibractea because it's the only species listed under the genus. Okay, our group proceeds is leading the global tree assessment of 800 Philippine endemic trees. I will discuss this in more details later. For us to determine how many native and endemic trees we have, we need to undergo a tedious review process. We search for available, reliable sources such as the protologs and monographs. They are mostly from very old publications, floras, taxonomic journals, and some online database. So based on our database, we have 3,283 native trees and 1,487 or 45% of which are only known to occur in the country. The families with the most number of native trees are Myrtaceae, Euphorbiaceae, and Moraceae, while Syzygium and Ficus are the most tree-rich genera. Syzygium alone has 195 species, which makes it one of the most complex plant groups. It's really very challenging to distinguish them from one another. Okay, almost 3,300 native trees. I hope everyone here knew at least 10% of these native trees. But the reality is, many of these species have never been documented for several decades now. And probably around 20% has no other documentation aside from the original publication. And considering the rate of forest degradation in the Philippines, we do not know if this species still exists. Based on the data of ESSC, we lost around 52% of our forest from 1900 to 1999. The data of FMB, as cited by Monga Bay, showed about 4% increase in forest cover from 18.3% in 1999 to 22% in 2007. The 2012 Philippine forestry statistics further showed 2% increase from 2007 to 2010. However, if we look at the quality of forest, we will see that the open forests were the ones increasing, while the closed forests significantly declined by 10% in just 3 years. FAO estimated that between 2000 and 2005, the Philippines lost 2.1% of its forest cover annually. And it represents the second fastest rate of deforestation in Southeast Asia, second to Myanmar, and seventh in the world. And based on the study of Conservation International, the Philippine forest is the fourth hottest spot in the world. In fact, we should be tied for third with Sundaland, with only 7% original forest left. And considering that trees are the main component of the forest, it is safe to assume that many of our trees were also gone with the forest. The updated red list for plants issued as Department DNR Administrative Order 2017-11 included 389 threatened plants, including 668 angiosperms and 21 gymnosperms. For trees, 347 were categorized as threatened, but take note that I included the OTS or other threatened species in the number. 347 or just 10% of the total number of trees. It seems quite low for a country that lost 93% of its original forest cover. Nonetheless, Dipterocarps has the most number of threatened trees. But how about in the IUCN Red List? I am glad to inform you that IUCN now has a partner in the Philippines for the Global Tree Assessment. EDC and BGCI, or Botanic Gardens Conservation International, signed a memorandum of agreement in May last year for the global tree assessment of 800 Philippine endemic trees. But how come EDC became the first and only partner organization of IUCN in the Philippines? Let me give you a very short background. So BGCI is the IUCN Secretariat and Coordinator for the Global Tree Assessment. The objective of IUCN is to have a red list assessment of at least 80% of all the trees in the world by the end of 2020. In 2018, there were only 141 Philippine endemic trees with IUCN red list assessment. Hence, they were really looking for partners and volunteers. The EDC, on the other hand, has a long ongoing CSR program called BINHI, which focused on the conservation of Philippine threatened trees. Since 2008, BINHI had been exploring and documenting remaining natural populations of these threatened trees as source of planting materials. With the successful search of the 96 priority threatened tree species in 2015, EDC extended its MOA with Biodiversity Management Bureau to include protection of their populations. This is the very first Adopt a Wildlife Species program for plants, as previous AWS projects were all focused on fauna. And under this MOA, another important task of EDC is to contribute in the updating of the conservation of the 96 species. Our more than 8 years of exploration in Binhi revealed several undocumented populations of our priority species. For instance, this mapilig tree, or Santostemon bracteatus, one of our ironwood species, prior to Binhi was known to occur only in Samar and Camarines Norte. But we were able to document it in 10 other provinces, including Aurora, Nueva Ecija, Albay, Catanduanes, Leyte, Dinagat, Surigao del Sur, Davao Oriental, and Compostela Valley. The same case for Bago Adlaw, or Santostemon philippinensis, which was known to occur only in Samar and Paracale, Camarines Norte, the gold capital in the Philippines. We encountered several populations of the species in 3 other provinces, including Isabela, Aurora, and Surigao del Sur. And results of our inventory also showed extreme abundance and dominance of the species in their area of occurrence. This is a natural stand of Malabayabas, Tristaniopsis decorticata, in Aurora Memorial Protected Landscape. Malabayabas is categorized as critically endangered in 2007 Red List. But our inventory estimate in just this one mountain already exceed 15,000 individuals. Here is a top view of the almost monotypic stand of Malabayabas. Same species, but multi-layered. We were also surprised to learn the very wide range of elevational distribution of Malabayabas. Available literature suggests that the species is a low elevation species, but in our traverse in Mount Mingan in 2017, we witnessed how Malabayabas is over-dominating the forest from 300 meters above sea level all the way to the peak around 1,700 meters above sea level. This is Palawan Mangkono, a province endemic species and also categorized as critically endangered species. But in Palawan, no one will believe that the species is a threatened one because it is the most abundant and the most common species in the province. In mainland Palawan, the species is widely abundant from Bataraza, the southernmost part, to El Nido, the northernmost part. And this photo is in Magarwak Forest in Puerto Princesa. And you can see a huge natural monoculture stand of Palawan Mangkono. All the islands in Calamianes group of islands, except Coron, which is pure limestone, are also dominated by Xanthostemon speciosus, which is locally known as bungan. So we explore every island of Calamianes, Apo, Dinanglet, Busuanga, Kalawit, and Culyon, among others. And we found out that the dominant vegetation is a savanna-like forest, dominated by grasses with scattered trees. In this vegetation type, about 90% of the trees is bungan or Palawan Mangkol. So with our extensive explorations of the populations of our priority species, best position to assess and determine their conservation status, because all the criteria for assessment requires knowledge of species population in the wild, population size reduction, extent of occurrence, area of occupancy, number of matured individuals, and probability of extinction in the wild. Okay, so we started our assessment in 2017. But because of the very tedious process of IUC and red list assessment, we just started with 5 Xanthostemon species. And our assessment suggested downgrading the 5 Xanthostemon species. Again, this is mainly due to several new populations recorded and the discovered abundance of the species in their areas of occurrence. Our assessment passed the review of BGCI experts in 2018, and it was published in the IUCN Red List as part of the 2019-1 version. Same with my feelings for very few Philippine species published by Filipino taxonomists. It's very satisfying to see that a Filipino firm is now cited as assessor of Philippine endemic trees. Previous assessments of Philippine endemic trees were all credited to World Monitoring Center or some foreign botanists, some of which have not even visited a forest in the Philippines. So, recognizing the exploration works of EDC and its capability to complete IUCN Red List assessment, BGCI partnered with EDC to conduct the GTA or Global Tree Assessment of 800 Philippine endemic trees. To date, we already submitted assessments of 491 endemic trees. 45 species were already published in IUCN Red List as part of version 2020-2. The remaining assessments will be published this first week of October as part of IUCN version 2020-3. So of the 491 trees we assessed, 455 trees are threatened, but this includes near threatened. And 183 of which are critically endangered. These numbers are far above the threatened trees categorized in the updated Philippine Red List, which is only 113 and only 36 critically endangered species. The families with the most number of critically endangered species are Myrtaceae, Lauraceae, and Aricaceae. Most of these species could easily qualify for the critically endangered category because of only one known location. And usually it's the locality where the species was discovered. The only reference for many of these species are herbarium specimens, usually holotype or isotype. And due to rarity and lack of reference for identification, conservation of this unknown species is a very tough challenge. Speaking of conservation, the most logical strategy to bring back the forest, as well as the abundance of our native trees, is of course reforestation. And the Philippines has a very long history of reforestation, which started as early as 1910. The most recent national reforestation initiatives include the Community-Based Forest Management Program from 1970 to 2010, the National Greening Program from 2011 to 2015, and now the expanded National Greening Program. Unfortunately, our old planting practices resulted to further degradation of our biodiversity. In fact, reforestation in the Philippines can be considered the second greatest cause of species degradation next to deforestation. Our reforestation in the past can be best described as planting by convenience, characterized by strong bias towards the use of exotic species, which were highly favored because they were the ones readily available in the market. These exotic species were planted in almost all protected areas in the country, leaving very limited space for our native trees to grow. And our reforestation programs has a production mentality. Our planting is always equated to harvest and profit, neglecting the main purpose of greening and rehabilitation. This is a relatively recent DNR administrative order. It is clear, or it is clearly stipulated in the basic policy that this is in support to the trust of the government to conserve the country's forest natural resources. But the appendices of this DAO showed uh, this uh, this policy that crooked stems and trees with forking stems are unacceptable for planting. If you are doing coastal rehabilitation, how come these natural beach species, which are naturally forking and with crooked stems, became unacceptable? In fact, these characteristics are the best to control for coastal protection against waves and winds. And our old planting has no consideration on the ecology of the species. This is one of the reforestation areas I assessed and evaluated in the past. This is in Calawi, Thailand. The area was planted by the exotic Acacia magium. Planting materials came from the mainland Luzon. So imagine the stress incurred by the seedlings during the transport and of course the transport expenses. Ironically, during our visit, we had documented a lot of native species flowering and fruiting in the area. These naturally occurring species were proven to be adapted and suited in the site, and therefore the best or the most suited for planting in this area. And our government has the plant now, buy now mentality, ignoring the basic silvicultural processes in planting, which should start from identification of mother trees, collection of seeds, propagules, establishment of nursery, and hardening of prior to planting. During the first quarter of the implementation of National Greening Program, there was already a target of hundreds and thousands of hectares to be planted. The field offices therefore resulted to buying readily available planting materials, which were mostly exotic species. And most planters were very good in publicity reforestation. Politicians, students, and other sectors, sometimes showbiz personalities were invited in planting activities to project successful participation in reforestation project. But after some photo ops, the main purpose of the activity, which is growing seedlings, will be forgotten. It should be emphasized that reforestation is just 10% planting. The bulk of the task is the nurturing of plants. These old planting practices should be removed and eliminated for our reforestation and tree conservation to be successful. We need to make the right practices as the new normal in planting. We must have bias against exotic trees and mainstream planting of native trees. We must remove the production mentality, but instead make conservation a default objective of planting. Planting of native trees could create a healthier ecosystem for both wildlife and humans. And as native trees are found in natural forest, the government should allocate funds for seeds and wildlings collectors for the establishment of nursery. This could be more than 10% cheaper than buying seedlings. Aside from ensuring continuous survival of native trees, it could also provide livelihood to upland communities. And lastly, we must forget the old practice of plant and forget. Seedlings need our care at least in the earlier years of development, especially in marginal areas, which are the usual targets of reforestation. No tree seedling can compete with kogon and talahib, and therefore regular weeding and tending is necessary. But apart from shifting the new normal to the new normal in planting, the government must strengthen forest protection. In one of our tree planting activities in Binhi, while we were planting at the school grounds, we were hearing the sound of chainsaw from the adjacent forest. So is it a lot of money to bring back the gun forest? We are at the same time allowing illegal loggers, poachers, and kaingineros to take down the our remaining forest resources. If we can protect our forests, then reforestation will be an eternal task of the government. This is, yeah, that's why forest protection must be a government priority. This is a perfect example uh, of, of that case. The UP Mountaineers adopted a deforested area at Ipo watershed in 2007. And after 6 years of planting and nurturing, they successfully reforested the site using pioneer native species. But in a glimpse, that forest returned to a barren site in 2014. As discussed earlier, our country already lost 97% of our forest cover. There were more likely a lot of undescribed species that were already gone even before their discovery. This figure shows the diversity of organisms on Earth and the estimated number of undescribed species per group. So for plants, there are still 40,000 species waiting to be discovered and described. So while crises on biodiversity degradation have been widely recognized, there is a bigger crisis that we should address. This is the crisis on declining number of people studying biodiversity. We should remember that we owe the taxonomists all our knowledge on biodiversity. Without them, we have no species to talk about. Filipino taxonomists are probably the most threatened species on the planet, even more threatened than our Philippine eagle. Here is a photo of our meeting last 2015 in trying to revive the Philippine Flora Exploration Project. We realized then that we were only about 20 people working in plant taxonomy in the Philippines. In the same meeting, we talked about the problems, why there were very few people getting interested in biology. The answers were quite obvious. Biology is not a popular field, and people perceive that being a biologist, unlike being a lawyer, a doctor, or engineer, will not make you rich. But you know, life is not all about making money. It's about doing the things that makes you happy. I may not have much money, but as a botanist, I always feel I am richer than any lawyer or doctor or engineer. It's because of my very rich experience and intimate relationship with nature. Discovering new species is priceless, and conquering unexplored mountains worth no amount of money. I feel so blessed and extremely satisfied that as a botanist, I am being paid for a job I wanted to do most. That's why I am challenging all the young ones, especially the millennials out there. Leave your computers for a while, walk through the forest, and interact with trees and wildlife. And I'm sure you will experience a different connection with nature. Thank you for listening.
Thank you very much, Professor Malabrigo, for that enlightening. To remind our audience, if questions for either of our speakers, Please write them on the chat box and they will try to answer them after the end of the second presentation. Now, on to our second speaker. Dr. Lerma San Jose Maldia also obtained her bachelor's degree in forestry from the University of the Philippines, Los Baños. She then went on to finish her master's and doctorate degrees, both in agricultural science at the Nagoya University Graduate School of Bioagricultural Sciences in Nagoya, Japan. She has since returned to UPLB, where she is currently an associate professor. Her areas of specialization are in evolutionary biology, conservation genetics, and tree genetics, particularly population genetics, phylogenetics, and phylogeography. Her talk today will be on molecular data in the conservation of forest genetic resources of native trees. Please welcome Dr. Lerma Maldia.
Okay, can you see the slide now? Thank you very much for that introduction. So, napaka-interesting nung sinabi talaga ni Prof. Malabrigo, no? But for my topic, what you will learn from this talk will be, I'll just be giving some overview of the relevant policies and measures related to conservation and some of the approaches in forest conservation. Then I will be sharing some of our current initiatives when it comes to generation of molecular data. And I took some documented related studies. And during the presentation and throughout later the presentation, I will be giving some implications of these molecular data for the conservation of native trees. So I think the diversity of the Philippines has been emphasized already by Prof. Malabrigo earlier. But here I just want to emphasize that in two issuances of the Philippine Plant Red List, you can see that the threatened plant categories are not really decreasing, but decreasing almost doubly. And in the Philippines, we have a lot of legislative and policy measures. So to some of the participants who are knowledgeable on this, so you know that we have a lot of legislative and policy measures related to forest conservation, related to conservation of biodiversity. So I just pick up some of them. I think are the most relevant. We have the Forestry Reform Code, the PD 705, so that effectively conserve the country's public forests or provide major framework for the management and conservation of forest resources. So the most widely tool we are using for our in situ conservation, the National Integrated Protected Area System. And I think you are all familiar with the EO 247 on the bioprospecting law that prescribes the guidelines and establishes regulatory framework for bioprospecting of biological and genetic resources. So whether it is for scientific, commercial, or other purposes. So the conservation of our biodiversity is also bound by an international agreement. And I think you're familiar with the Convention on Biological Diversity. So foremost objective of this international agreement is to conserve all components of biodiversity. And these components include the genetic diversity, the species diversity, and ecosystem diversity. So you can see here in the hierarchy of biological diversity, the base represents the genetic diversity. So this is the simplest, the smallest scale among the biological diversity, but it is the foundation of all of all biodiversity. Um, and if you are familiar with this country report by the DNR on the state of the forest genetic resources of the Philippines, so this is our contribution to the state of FGR in the world. So it was reported that we are implementing both the ex situ and in situ genetic conservation. So among the ex situ, for those of you who are familiar with, so we are implementing sound field gene banks, botanical gardens. I know you are familiar with this. arboreta, so clonal propagation, so for the establishment of clonal seed orchard, which later can be source of improved seeds. We have the seedling seed orchard and those seed production areas. However, we have not yet implemented some of the recent uh, advanced technologies when it comes to genetically ex situ genetic conservation. You know? So in terms of forest seed banking, for example, I know that the ERDP-DNR is pioneering on the seed storage of limited numbers of orthodox seeds through the Mindanao Tree Seed Center. But this one is an example. This is an FAO-initiated seed bank, the Millennium Seed Bank Partnership, which aimed to store forest seeds of forest trees and other woody plants. So India, for example, has already invested a lot of money in their collection of seeds from all germplasm, although for forest trees, their initiative is still at its infancy. But they are already advanced in the in vitro gene bank. So it makes use of micropropagation techniques to conserve all the available genetic variabilities. So they are also now into cryopreservation, so making use of the liquid nitrogen up to negative 196 degrees centigrade to conserve for plant species. Normally, it's the dormant buds, nodes, or young stems, pollen, ovule. So all of these things can be already preserved using this particular technology. So I think that will be a big challenge for our government to really put money into this kind of conservation. But as mentioned, on one hand, on the in situ conservation, so Prof. Malabrigo already mentioned a lot of the initiatives when it comes to um, in situ genetic conservation. Okay. So, and I think Prof. Malabrigo is really, really has been it's part of this particular initiative by the government, by the DNR. No? We have the Plant Committee on conservation, and this is we have the 10-point plant conservation strategy and action plan, which aims to halt the loss of plant diversity. And I think in those, among the 10 strategies, generation of molecular data to assist conservation falls under these particular strategies, particularly strategy 3 on the promotion and support of research on the genetic diversity, systematics, taxonomy, ecology, and conservation biology. So, okay, and here, what I'm showing to you are techniques to, sorry. So here, what I'm showing to you are techniques to generate molecular data. So molecular data techniques have already, since the past few years, techniques to generate molecular data has advanced greatly. So we have, if you are hearing genomics or the omics technology, so genomics, which normally involves the DNA, transcriptomics, proteomics, metabolomics. But for the purpose of this particular presentation, I will be focusing only on molecular data that are generated from variation in nucleotide sequence or variability at the DNA-based molecular marker. So malayong-malayo pa yung ating pupuntahan for forestries dito sa ating sa mga omics technology. Ayaw mag-enter. Sorry, ayaw mag-enter. Okay. So, molecular data, here in this slide, you can see that molecular data are generated from variation in sequences. So these sequences and this variation in sequences can be used for species discovery, particularly in the case of the DNA barcoding. So it can support species identification. So in the absence of very distinct morphological characters to species, then the use of variation of nucleotide sequences is a big potential to help in the species discovery. And also these nucleotide sequences can be used in, particularly those variation can be used not only in species discovery, but also in determining evolutionary relationships among species, and not only at the species level, but also among populations of a particular species. And here in the graph, in the photo, you can see that recently the even DNA barcoding is used for conservation. So not only for taxonomic purposes. And generation of molecular data can also be obtained from variability in size of DNA fragments among individuals at a locus or molecular markers to constitute genotypes. In this example, I'm showing you particular polymorphism or variability in microsatellites. So these are repeating units of the nucleotide sequences. Okay, and normally we can do sequencing, but in many cases we just do fragment analysis. And with differences in size, then it shows variability among individuals. So this is an example of our recent study on Almasiga, on Agathis philippinensis. So we sampled populations from Davao, Marufinas, and the Brooks Point in Palawan. And then we have representative population from Santa Maria, Laguna, which were presumed to come from Quezon. And then we have few individuals in Mount Makiling Forest Reserve. So in this particular, in the result, okay, before, so based on IUCN, the Almasiga, our Philippine Almasiga is synonym to Agathis damara. So this is vulnerable by the IUCN, and we have in Palawan we have bagtik. The locals called it bagtik or Palawan Almasiga. It's A. celebica, but it's a synonym of Era. And then in Davao Oriental, in Governor Generoso, locals have identified white and red type, but that is not based on the actual color of the wood itself, but based on the production of resin. So one is slow yielding and the other one is high yielding in terms of resin yield. Also in Palawan, we were able to ask the natives that they are also considering red almasiga as another type. And similar to Davao Oriental, that is also based on the amount of resin produced during the collection. So based on a certain gene, the MATK gene, the chloroplast MATK, we did not really see specific variation in nucleotide sequences. And the Adamara, but our samples diverged from Adamara. However, there is one particular sequence deposited in the NCBI for a MATK, and that one deposited sequence clustered with that Adamara. So in that case, this particular situation can be a little problematic. However, when we sequence the RBCL, another chloroplast barcoding gene, so with RBCL, you see here that the Agathis philippinensis, the one deposited at the NCBI, clustered with our sample. Because of that, we were able to see distinct differentiation between populations of the Governor Generoso and that of the Palawan, Quezon, and those from the Makiling Forest. So these are very interesting, interesting results. And this, although there are a lot of reported places for Almasiga. So, but based on our current samples, are these two distinct populations? Can we say already that these are already two distinct species? Because RBCL, their differentiation was only due to a point mutation at a single site of the RBCL gene. And so with barcoding, can we use, we are thinking if RBCL gene is an effective barcode for Almasiga. Okay. So another one is ironwoods. So Professor Malabrigo has already made mention about ironwoods, you know. So they are known for their exceptionally hard and high density wood. So in the Philippines, the Santostemon and the Tristanopsis are the known ironwood trees. So they are collectively known as ironwood trees, but they are currently under serious threat. So both by human and other biological causes. So in this particular photo, I showed you our initiative on documenting the damage, you know, biological damage, diseases in Malabayabas stands in Atimonan, Quezon. So this is our current project uh, funded by the UP system. So other than documenting, we tried to implement tree surgery for those individuals, particularly along the trains where people visit the area. So yun po yung isa dito na about Malabayabas. And one of our students sequenced the RBCL gene for samples from Santos Temon and Tristanopsis. So, okay. Sorry. In this phylogenetic tree, the genetic, you can see that X, the Santos Temon, and then the Tristanopsis. So this particular tree showed you the divergence of Santos Temon and Tristanopsis. However, when we use the outgroup from, for example, the Lithocarpus and Villeniacii, we still see, but we confirm the genetic relatedness. By evolution, we confirm the genetic relatedness of these ironwood species in the Philippines. Okay. When sequences were compared against when sequences were compared against the available ironwood sequences deposited in the database, so the genetic divergence of the two genera became much clear, placing the suspected Tristanopsis. So here you can see the Malabayabas puti. So the locals in Atamonan called it Malabayabas puti, although we have not collected the samples for further identification by morphology, but in this particular study, it placed the suspected Tristanopsis or Malabayabas puti under the Tristanopsis group. Okay, so when we compare that with the available sequences in the database. So, but to clarify this, other genes are needed to be analyzed, and genetic diversity underway. So genetic diversity within population will provide estimates at how much genetic diversity is maintained in the remaining populations of ironwoods in the wild. And so we have an upcoming DNR FAST project. So we are going to not only sequence genes, but we are going to look into the diversity of populations of Santos Temon verdugonianus, the one which is relatively widely distributed. So we are going to see how much diversity is present in Mancono. Okay, so this one is our MS, now an assistant professor in CFNR. He sequenced the Philippine dipterocarps. So, and these are our premium hardwood species, but sadly, in general, they are classified as threatened species, most of them. So sequencing the chloroplast gene, so clarified the genetic identities of taxonomically problematic species. So the red box that you see, these are some of the debated species. So at least we were able, so for this particular study, so we were able to show you that this problematic species can be resolved. But in general, what RBCL showed us is we were able to see the evolutionary relationships of genera of Philippine dipterocarps. So we we can see the Churea, Hopea, Dipterocarpus, Batica, Anisoptera. So there is clear divergence among separation among these genera. And also, this particular study can serve baseline data for species discovery and confirmation. So at this time, if you look at sequences of our Philippine dipterocarps in the DNA database, so you will rarely find the sequences. So this will be very useful. The sequences will be very useful for confirming the identities of our Philippine dipterocarps. So I think one of my students is attending here. So he did this particular small study on Hopea malibato and Hopea paxworthy. So these two particular species are by morphological identification. I think if you are not an expert, then it will be very difficult to really identify which one is malibato and which one is Hopea paxworthy. So he sequenced for partial of the MATK gene and the RBCL. But in the RBCL gene, we did not see any variation in sequence. So H. malibato and H. paxworthy were clustered, including the H. Hopea kisumbiniana. So, but when he sequenced the MATK gene, so there is one single 1-point mutation, So at this site, around 584 site on a partial sequence of MATK, he was able to, under my supervision, he was able to delineate H. malibato from H. paxworthy. So it's very interesting that at this particular level, at the sequence level, we can identify, in the absence of this flower, for example, that will give a better delineation for two highly related species, then molecular data through sequence, for example, is a very good one in delineating species. So currently we have an initiative on the Philippine tick. So we know that these are only limited in distribution in the Ilin Island and Occidental Mindoro and in Lobo and San Juan, Batangas. So we have initially sequenced the genes MATK and RBCL, but in terms of RBCL gene, we just confirmed its close relationship with the Tectona grandis. So now Tectona grandis is being widely planted, particularly in the northern region. But there was a reported study by Agoo in 2008, but this is also based on variation of sequence in the TRNL intron. So definitely, as expected, with the low polymorphism in nucleotide sequences, so haplotype diversity. So there is little nucleotide diversity between these two populations. But we have already started our population level analysis using the more variable markers. So this is to determine their fine scale genetic variability and structure. So currently, we have also we have started our initiative through the SLSU project. So on batikulin, so batikulin or let's say latency is an important is a preferred wood for wood carvings in particular. So we have, as expected, we obtained absence in the sequence of variation, but underway, we are going to do population genetic diversity analysis. This is to assist in the selection of stocks for the establishment of clonal seed orchard. So hopefully the phase 2 of the SLSU project will be granted. And then we have documented some initiatives already by our colleagues in the university and in the DNR. So this one is in Shureya Giso and also in Parashureya Alanonan. So in Shureya Giso, high levels of genetic diversity within there is higher level of genetic diversity within the population than among populations. But you can see here, so Dr. Tino was able to clarify that the Bohol population was the most divergent among the populations. So both natural and planted populations were included in the study. Although we can see that the Makili, the Nueva Vizcaya, Bislib, Subic formed into one cluster. On one hand, in a limited population from Leyte, so there was moderate amount of molecular diversity was observed. And then in the effort of the DNR for five important priority species, their priority species, this one is in Nara. Ano, you can see that although relationships of populations did not conform with their geographical location, then the differences in the color determines a particular distinct, we can distinguish one population from the other population. So it's a good thing that we can see some of this differentiation. So on the left, you can see that this is a reported study by Dr. Quimado of UPLBC FNR. So on seed production areas of Venus cassia or Benguet pine. So for of the four seed production areas, so there were two clusters that were observed. So of course, Benguet and Nueva Ecija, we can see some conformity in their geographic locations, but Ilocos and Bukidnon, so there could be some translocations if there is the possibility of translocation because of their geographic differences, napakalaki. And at the level of individuals, we can see similarity, okay, in their genotypes. So this one, the axis 1 and axis 2, you can see that they share some of the individuals from different seed production areas. Okay. Um, so these results, so the ones I showed you in this slide, so these results on variability and differentiation of populations are very important to understand in the selection of provenances to be used as seed source for ex situ conservation, such as establishment of seedling seed orchard or field gene banks, where genetic variation of genetic materials is a prerequisite. Ano? So how can molecular diversity be made useful for genetic conservation? So there are two aspects. So of course we need that the genetic diversity must be maintained at a considerable level necessary for their short-term ecological adaptation. So knowing the levels of diversity in a species is very important in determining which conservation measure to take. So it is the genetic diversity that determines the potential of a species or populations to adapt to changing environmental conditions. So in case where genetic diversity is low within a population, then it can threaten their long-term persistence. So I took these guides on when to implement a particular conservation action, when to take an ex situ and when to take an in situ, or there should be just a status quo. So with information on genetic diversity of a particular species, so for low variation within a population, for example, in an isolated population containing very less amount of individuals, it's a prerequisite that an in situ conservation must be complemented by an ex situ. So in the case where there are fragmentation among populations, so whether it is genetic fragmentation or the physical fragmentation, then an in situ conservation is needed, particularly if populations possess some kind of local adaptations. It will be very, according to by genetics, if there is local adaptation, then their translocation to different habitats will be detrimental to the population. And so in a large contiguous population, so if genetic variability is okay, so it is sustained at a certain level necessary for long-term persistence, then there should be status quo. So in the event that this is a good discussion, so we with limited resources. For example, our government has limited resources. So we can decide which particular population is to be conserved and which particular conservation action is to be put in place. So in summary, accuracy in identification of biological organisms, genetic information on levels of diversity, and structure of populations of a species are prerequisites of conservation. So in general, molecular data of Philippine native trees, when they are made available and accessible, will be very important not only for purposes, but them from further exploitation and for facilitating the establishment of appropriate scale and even the strategy of its conservation. So, thank you. I-acknowledge ko lang po ang CFNR, UPLB, the DOST, and the DNR, my advisors and colleagues who are now, I think, watching and attending this seminar. Very much.
Okay, thank you very much, Dr. Maldia, for another fascinating lecture. We will now read questions from the audience. So our first question, are there science-based policies that arose from your research works? What are these? So for both speakers. Who wants to start?
Yes, I
Sir,
can.
pwede ba ito? Sir, pwede ba ikaw muna?
Yeah, I can start.
Yes, sir.
Regarding science-based policies on uh, our research, of course, with our BINHI project, so the updating of the conservation status, particularly in IUCN, is a science-based policy assist because, of course, when species is included in the red list, it would be a protected species. The same is true for the Philippine Red List. We are also coordinating with the Philippine Plant Conservation Committee for the next updating of the Philippine Red List. It's supposed to be updated every 5 years, but the last update went to 10 years from 2007 then 2017. And hopefully, our assessments, our updated assessment would also be included in the updated Philippine Red List. And with regards to the mainstreaming of native trees in planting, we already have a lot of partners, LGU, NGO, who are advocating the planting of native trees. In fact, a lot of municipalities had their municipal ordinances, some have their provincial ordinances to conserve the threatened species in their area. So these are some of the science-based policies that could influence the conservation of these threatened trees.
Okay, thank you, sir. Dr. Maldia.
Okay, thank you. So I think at the moment, information on genetic diversity as well as molecular data is still limited for use by the government because there are only limited institutions or research institutions. But of course, I mentioned the initiatives of DNR, but I think this is not yet incorporated in our strategies and even in our policies. But in my experience, with in Japan, so we had a very big project on genetic diversity assessment. So now those information are now being used in reforestation, for example, in conservation. So whatever patterns of genetic diversity. So if, for example, the northern population is different from or genetically different from the south populations, then the reforestation should not take place if it involves translocation of propagules. So I think for that particular experience, because in Japan, they are now using this information in the management of their genetic resources. So I hope DNR will also look into the possibility of getting information as provided by molecular data in their in setting up national strategies. Did I?
Okay. Yes. Okay. Thank you, Dr. Maldia. So for our next question, I guess this is for you. May you enlighten us what are haplotypes and microsatellite DNA markers?
Okay, sorry. So the microsatellite markers, so the ones I showed you with the repeating units, basically microsatellites, these are just repeating units of nucleotide sequence. It can be in a certain fragment, it can be a repetition of a repetition of TCG, for example, or TCG, 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 and that variability is so polymorphic across individuals within a population or populations among species. And it can detect even the finest scale of genetic variation. That's why up to now, since the development of that marker in 1990s, so this has been used both for plants and animals to locate the fine scale or to identify the fine scale diversity. In the case of haplotype diversity, it's just variation in haplotype. So in plants, mitochondria and chloroplast genes are haplotypes, especially for angiosperm. Haploid, yung N or diploid. So kapag ka-diploid, it has already the parents, magkasama na. So meron na siyang composition of the mother and the father. Kaya diploid. Yung zygote is a diploid. So kaya haplotype lang. When we mention, we consider haplotype because in angiosperms in general, mitochondria and chloroplast genes are inherited only from the mother. Maternal sila. Kaya haploid type. Ayun. Okay po ba?
Okay po,
So,
ma'am.
hindi ko na naiisa-isa kanina.
Okay. Okay, so the next question, aside from deforestation, what are the other factors that affect plant diversity or factors that make some plant species endangered?
Okay, probably the other reasons aside from, or the other factors, causes of the extinction of species or making a species endangered, aside from deforestation, is the proliferation of bioinvasive species. Because as you could observe, a lot of bioinvasive species are dominating our natural forest. It already intrude our natural forest. The likes of paper mulberry, buyo-buyo in Visayas and Mindanao, and even African tulip or Spathodea campanulata, which was introduced for landscape purposes. Now they are competing with our natural species in our natural forest. But aside from that, I think one of the uh, causes of extinction or endangerment of our species is, of course, the lack of awareness of the people regarding threatened species or regarding the identities of species. In fact, in many of our explorations, we have observed a lot of small backyard farms planting their preferred agricultural or fruit trees, but clearing up the area wherein the area has a lot of threatened species in our priority species list. So it's because they do not know how much or what's the value of the species inside their area. So they tend to clear it up and plant for less valuable species, at least ecologically. So we must uh, we must have an awareness campaign about this. And again, the lack of people who should make us understand the diversity of the Philippine plants, no? And those are the taxonomists. No? We need more taxonomists to understand our Philippine trees, our Philippine plants better.
Okay, thank you very much, sir. Now,
Yung,
another
pwede ako
question.
umako?
Yes, ma'am.
Actually, gusto kong mag-add doon, ano? So, doon naman sa aspect ng genetics, ano? So, with all those deforestation, ang nangyayari kasi, there is also a corresponding fragmentation in populations of a species. Kapag nagkaroon na ng fragmentation, there definitely, alam natin for plants in particular, nagkakaroon ng genetic diversification through pollination. Ano, so kapag ka nagkaroon ng fragmentation in their habitats later on, these individuals of the same population, supposedly initially of the same population, they are not permitted to have gene exchange. So kaya ang nangyayari, tapos maunti pa yung individuals dun sa fragmented populations, nagkakaroon tayo ng tinatawag na inbreeding. So sila lang yung nagkakaroon ng mating, and through the generation, then alam natin sa halaman, sa hayop, sa lahat yan, pag merong inbreeding is also, it can lead to extinction kasi iisa na yung kanilang genetic makeup. So yung similarity in genetic makeup is not really a good indication. Kasi pwedeng kapag may dumating, for example, there is one particular natural disturbance or biological disturbance like disease, so pwedeng ma-wipe out yon because they only contain similar genotype. So siguro yun ang may dadagdag ko doon, but sa genetic point of view.
Thank you very much for that, Dr. Madia. Minsan nakakalimutan natin may may contribution din ang molecular data sa ating mga conservation and reforestation efforts. Yes. So the next question is again for you. So from this molecular study of agave species, is it conclusive for us to say that A. philippinensis is not only a synonym of Damara, but they are of two distinct species?
Okay. So based on the data I showed you, of course, we are still verifying a portion of the MATK. So MATK gene is very long. It's really long. We only sequence 2 markers to connect the partial MATK, and we are confirming whether there is no variation between for this particular between Damara and Agathis philippinensis. So by RBCL, we can say at least that at this time, hindi pa naman talaga ako magbibigay ng conclusion at the moment in final, but the synonymity of A. philippinensis may be nullified. Kasi we have already sampled populations from several individuals from populations in the Philippines for our Agathis almasiga, and we were able to show na there is differentiation in populations, pero na-separate natin sila with A. damara. So that is with the RBCL.
Okay, thank you very much.
But we will confirm that we are now in the population genetics of almasiga.
All right. And the next is, on lapnisan or agarwood, do you have studies on its molecular properties and geographic distribution in the Philippines?
Wala pa po, but in our September presentation, ako po si Prof. Malabrigo and a colleague in UPLB, we were granted a project by the DOST. So yun po ang aming kakaharapin in 2021. So makakaasa po kayo, it will be presented.
Okay. So the next is, how can we encourage today's Filipino youth to take up botany, forestry, and or conservation related courses? Thank you po. God bless.
Okay, I think it should start from probably from the elementary years, because I read a study that our textbooks in the primary and even secondary education really lacks introduction or giving appreciation to our biodiversity. So it all it should all start from interest, no? And you cannot have an interest or passion to a thing which you do not know. So we should increase our awareness campaign regarding the importance, the diversity, as well as the importance of our Philippine biodiversity. Right now, we are, that's why the target of our BINHI, the target partners are usually schools, primary schools and secondary schools. And we don't just plant the threatened trees, we also put labels containing important ecological and interesting information about the tree. In that case, we are hoping that the students could have some interest, or some students would be interested in knowing more about these Philippine trees.
All right, thank you. So there is a comment here. So I guess this is for people asking for copies of your presentation. The presentations done today by resource speakers will be of great support if can be presented in my classes. So would you be willing to provide copies of your presentations, or can they can just watch the videos again on YouTube and our Facebook page?
It seems they are asking for the presentation for sharing to their class. So I think we can give them the copy as long as proper citation should be done during the presentation. They cannot claim it's their presentation. So it's just like that, proper citation for the authors.
Yung sa akin naman, yung presentation kasi, it includes mga
my
data.
data.
It is,
Yes.
yung data kasi I already presented in conferences, but pwede naman din yun kung i-ano lang, yung titingnan lang. So okay naman yun, but not for distribution to kasi isusulat pa lang yung iba doon. Okay? Thank you.
Okay. Sige. Tama. So another question, are you planning to have lectures on how you are doing different in situ procedures in preserving biodiversity?
Ako, Sir Pat, ikaw ang, si Sir Pat ang talagang po natin.
Lectures on in situ conservation? Is that the
Yes,
question?
in situ procedures in preserving biodiversity.
I haven't done it yet, but I think we can, if invited, we can share some our experience in dealing with the in situ or protection on site of our priority species. But I can say that a good formula should be a collaboration with as many possible stakeholders in the area. The LGUs, the DNR, the NGOs, for the conservation to be really successful.
Okay. Yung kasing in situ conservation natin, widely or majority ay yung protected areas. So it is the DNR, kumbaga, yung sa kung we can provide some lectures, but yung DNR yung talagang mas nasa mandate niya, yung mga protected areas. But for the ex-situ, we have experiences in the ex-situ conservation, then yun siguro kaya kong maibigay yung mga yun. I can provide lectures for the ex-situ, yung mga nabanggit ko kanina.
Okay, thank you. And well, sige, let's see how many more questions we can accommodate. So this next one is, being experts in forestry, how can you address inappropriate way of reforestation? Example, mahogany, Jumelina reforestation, a threat to evasion in local forest communities.
Sige, Sir, ikaw muna.
Okay. So again, aside from a lot of information dissemination campaigns that we're doing and piloting native tree arboreta, we need we really need to have an enabling policy that should support mainstreaming of native trees. It could be local, it could be provincial, or it could be national. But in fairness, now these days there is the past decade maybe there is an unprecedented increase in the appreciation of native trees. In fact, everybody is now talking about planting native trees. Papagalingan pa sila kung sino yung mas maraming naitanim. No? And not only for the private practitioners or enthusiasts, but even the government. I learned that expanded national greening program now uh, is trying its best to really use native uh, species for planting. So yes, enabling policy should be formulated.
Okay, yung sa sagot ko dyan, in many cases, kapag nai-involve ako particularly in tree planting, as much as possible, dahil nga nasa line tayo ng genetics, lagi kong ini-emphasize that in any reforestation, for example, or in any ex situ conservation, we must introduce variability in the sample. So, hindi po pwede na although tayo ay gumagamit ng cuttings for mass production, so ang dami natin, we can provide seedlings from cuttings from thousands of samples. But if that is only, if those cuttings were only from one mother tree, for example, one parental tree, so it is not that good. So kaya ang ginagawa ko, as much as possible, I try to incorporate yung mga information on the importance of diversifying your materials. So hindi naman masama that you use cuttings, for example, in reforestation, yung mga samplings, but as long as these cuttings came from several parental trees, then meron ka pa ring diversification. So yun ang lagi kong sinasabi sa mga, and also in the selection of parent materials ng new seeds. Hindi pwedeng sa isa lang kayo kukuha and then you produce thousands of seed from that single tree. So lagi kailangan there should be diversity of the seed sources para ma-maintain natin yung variability na nandoon sa i-introduce natin. Kung sa isang area yan, kung sa isang denuded area and we are going to introduce natives sa ibang lugar nakikita, at least we still provide some diversification ng ating mga planting material. So, yun po ang lagi kong sinasabi.
Okay, thank you very much. So there are still a few questions, but unfortunately, that is all we have time for. So the questions that were not answered today will be given to our speakers and they will respond to you by email. So again, thank you very much to our speakers, Professor Malabrigo and Dr. Maldia, for your very informative lectures and very inspiring. And thank you everyone for attending the webinar and for participating in the Q&A. So we hope you have learned a lot from the lectures this session. So may I now ask everyone to turn on your cameras for the photo documentation. Picture. Sandali lang po. Medyo marami kasi tayo. Tagal naman. All right. Thank you again for that. So before you go, audience members, before you go, the link you now see on the chat box is for the evaluation form. Kindly accomplish it to receive your certificate. This link will close in 15 minutes, so we hope you can answer them soon. Right? And please also visit our Facebook page for the announcements of the next lectures. So have a good day. Stay safe. Ay, wala pa pala ang link. Link, please.
Ngayon, ikaw magbibigay ka ng gagawa? Oo. Ano ka ikaw?
There. Nakikita nyo na po yung link. Okay. Thank you everyone for coming.
So,